Hey guys, welcome for chapter 11, cell communication. Now, this is a chapter that we really need to focus in on. We need to be willing to ask questions on because this is likely something that you've never heard of before or definitely never learned about. So, cell communication still very important. And this is it's a big topic for the AP Bio test. So here we go. Some things to think about. Why do cells need to communicate? How do cells communicate? What are some examples of communication with cells? So, this is an absolutely essential part of a multicellular organism. We have all kinds of different cells. If you think about all the different cells in a multicellular organism, it's very numerous, okay? But they need to be able to interact and communicate with each other. For instance, one part of the body might make a hormone that affects the rest of the body or another part of the body specifically. So it is very important that cells can communicate with each other. And they do this usually through chemicals. And again, they're coordinating the whole body's activities. So we have discovered as biologists some universal mechanisms, which is really how we will break this chapter down. 3 specific mechanisms. So section 1 is really an overview. So the overall look at this is how external signals are converted into internal responses within the cell. So how do cells basically respond is kind of the overall picture. So if we look at some really primitive cells like yeast, this is how they can reproduce. You can see here 2 different yeast cells. We have an A cell and an alpha cell, and they're going to exchange some factors. Okay, that's what you're seeing here. An alpha factor is being interchanged between them. And what you're creating is an A alpha cell. So these 2 cells have communicated via chemicals with each other and made a new cell. Signal transduction pathways convert signals on the cell's surface into a cellular response. So you see this pathway here. It starts on the cell's surface, which would be the cell membrane, and can result in a response by the cell. And amazingly, even though the last common ancestor for yeast in mammals was over 1 billion years ago, they're very, very similar in most organisms, these mechanisms. So cells in a multicellular organism, we're going to communicate via chemical messengers, also known as signal molecules, sometimes known as a ligand or ligand. But they can communicate both locally, which would be close by, right? And long distance would be over far apart, these cells. Maybe we can, for instance, have cells communicating with hormones that were produced up in the head region that go all the way down to your feet. So these signaling molecules are going to target other cells. So there's a couple different types. The first type we'll talk about is juxtacrine. And this is the key part here is they are directly in contact with each other. Okay, so you can see here, this is a gap junction. This would be plasmodesmata, but basically what you're seeing here is these 2 cells are in contact with each other. They have tiny openings in the membrane that separates them. So they can directly convey messages between them in that way. But juxtacrine means they're touching. So this is still an example of juxtacrine. So they're communicating again via direct contact. So again, this would have to be very local, very close by each other if they have to touch, obviously. So this is again an example of juxtacrine. But this cell is in essence conveying a message to this cell, and you can see it's coming into the cell. This type of signaling is especially important in early development. If you think of the development of a baby and all the different changes that cells have to make. Maybe one part of the body is going to become the liver, another part's going to become the brain. Okay, this is actually how they communicate early on in that. It also is very important in the immune system, which we'll look at a little bit later on in this unit. Another type of signaling is called paracrine. And paracrine signaling, this is more of a still local. It's going to be definitely farther apart than juxtacrine because they have to be in contact for juxtacrine. But paracrine is still local. So this cell, and if you look in this picture, it's sending out messengers, but it's still only sending these messages very locally. Great example of this would be neurons. So if you ever learned about action potentials and how they are sent, or with neurotransmitters that are sent across the synaptic cleft, that would be an example of paracrine. Very close, very local. Growth factor is another example of that. All right. And endocrine is the last type. So endocrine is associated with long distance. So you look here, how this is being shown is this cell is secreting this message into the bloodstream, and through the bloodstream it's being transported throughout the body, often associated with hormones. Hormones travel throughout the body through the bloodstream. Okay, so the endocrine system, it's a body system that creates hormones. How do they travel through the bloodstream? This would be long distance. All right. There's 3 stages of cell signaling, cell communication. You're going to hear this a lot this unit. Reception, transduction, response are the 3 types. And Earl W. Sutherland is really the scientist that figured this out. And how he figured this out is actually kind of interesting. What he figured out is the hormone epinephrine. What it does is it basically activates an enzyme. The enzyme is called glycogen phosphorylase, but you don't have to know. Just think epinephrine is a hormone and it's activating an enzyme. But what he did was he put those two in a beaker or a test tube together and nothing happened. In other words, epinephrine did not activate this enzyme when they were together in the beaker. However, it does activate it in an intact cell, meaning a fully grown cell. So what he basically concluded because of that is there had to be something on the plasma membrane that was involved with this. And he called that process reception. All right, so here is that signal molecule. This would be the epinephrine from our example. It has to interact with a surface receptor, and then it can activate the pathway, activate the enzyme in this case. Okay, but what you're seeing here, here's our ligand, or our signal molecule, our chemical messenger. It's bonding to a receptor. Sometimes that's on the cell membrane. We'll look at a couple different types of those in a little bit here. So that's reception. The signal molecule binds to the receptor and really initiates the process. The next step is transduction. So what we have here is a relay. There's going to be some amplification. There might be some secondary messengers involved. But in essence, it's going to bring that wherever it needs to go in a cell so that we can create a cellular response. All right, so the first part we're going to look at here is reception. So reception is when a signal molecule binds to its receptor protein, causing a change in shape or a change in configuration. So the target cell has a receptor, and this is a protein molecule, usually on the cell membrane, could be inside the cell. Again, we'll look at that in a minute, which can only recognize a specific ligand or signal molecule. They have to be complementary, meaning they have to be the perfect shape for each other. The binding of the signal molecule and the receptor is highly specific. If we think of the reason why, because proteins have a specific shape. They only fit that one specific signal molecule because of their shape. And that signal molecule, that ligand, creates a conformational change. What does that big word mean? It just means a change in shape. So the receptor is going to have a slight change in shape, and that's going to initiate the transduction of that signal. Sometimes we have intracellular receptors, and what that means is the receptor is not on the membrane. It is actually inside the cell. It could be in the cytoplasm. It could be in the nucleus. But in order to have an intracellular receptor, what we have to have is a signal molecule that can pass through the membrane. If the single molecule cannot pass through the membrane, that receptor cannot be intracellular. This is a big concept. It will be guaranteed to be on your test. So again, an intracellular receptor, one that is found in either the nucleus or the cytoplasm, if that's the case, the signal molecule has to be able to pass through the membrane. Otherwise, it can't get to its receptor. So usually, if that's the case, those types of signal molecules are small, they're hydrophobic. And if you think why, it makes sense, so that they can get across that cell membrane. So steroids would be an example of it. These would be non-polar. So you can see here's our signal molecule. It's going to pass directly through that membrane, and it's going to reach its receptor in the cytoplasm. It's still going to cause that transduction and a cellular response. It's just a different location for its receptor. On the other hand, a lot of the receptors are on the plasma membrane. The reason why is because their signal molecule likely can't pass through the cell membrane. So it has to have its receptor on the outside. So there's three types: G protein linked, tyrosine kinases, ion channels. I wouldn't spend too much time focusing on memorizing what each of these do, but I'm just going to go through a brief explanation of them. What we see here is a G protein. Again, here's our plasma membrane, our cell membrane. Here's our signal molecule. Okay, the signal molecule is activating this receptor, and what you're seeing here is it's going to activate a G protein, which is going to activate the next step. But again, what are we seeing? We have a membrane protein being activated by a signal molecule, which is going to basically trigger the next step. In this case, the next step is activating a G protein. Sometimes we have a tyrosine kinase, and a specific thing that is unique about these is they can create multiple cellular responses. But again, what are you seeing? Signal molecule bonding to a receptor protein, which is going to cause a change. The ion channel, this is one that is highly important because it's basically what we see in the nervous system and neurons. So, for instance, the signal molecule could be like a neurotransmitter, and that's going to activate a ligand-gated ion channel, which can allow, for instance, sodium and potassium through. When we look at an action potential, that'll make more sense. But what we're seeing here, again, we have a protein on a membrane. We have a signal molecule causing a shape change, which is going to cause a cellular response. If this was a neuron, maybe that could be an action potential. It could be a cellular response. All right, so that was reception, right? We had reception. The reception causes the next step, which is transduction. Transduction is a relatively complicated. I'll try to make it as simple as I can. Basically, we can produce a huge cellular response from a small number of ligands, small number of signal molecules, and it's kind of like falling dominoes. If you think about one step leads to the next step, leads to the next step, that's transduction. Key part about transduction is it amplifies the signal. So even though we might only have a small number of signal molecules, we can create a huge response. Great example of this. Our body can, in certain instances, maybe you just avoid a car accident, and your body is pumping with adrenaline. That is a hormone, but your body really doesn't need to make that much adrenaline in order for us to, our whole body seems to be alert and more focused, more powerful. And the reason why is because that small amount of hormone, that small amount of adrenaline can be hugely amplified in our cells. We don't need to, it also allows cells to not, or organisms to not have to work so hard. You only have to make a small amount of hormone to create a huge change. So at each step in a pathway, a signal is transduced from one form to the next, creating usually a conformational change in a protein. A lot of times that takes place by phosphorylation. What is phosphorylation? Just adding a phosphate. Now, it's important to remember that the ligand is not, the signal molecule is not entering the cell in this case. It's just activating a receptor, which activates the next step. Many signal pathways include phosphorylation cascades, which is basically just moving a phosphate from one part to another, and that activates the next step. And also, once I get to the picture, I think I can explain this a little bit better, but let me just get through a couple slides here. In this process, a series of protein kinases add a phosphate to the next one that activates it and inactivates it, and it's basically like a domino effect again. So here's signal molecule activating a receptor, that's reception. This whole process from that right in here all the way to here is transduction. Now, picture this as a domino. Domino activates the next one, activates the next one, activates the next one, until we get to a cellular response where we needed to go in that cell. So again, we started here with reception. That's going to activate. Now, what's happening here? You see here is ATP. Okay, so we've activated, we've phosphorylated it. That changes it, which activates the next one, and then activates the next one, and then activates the next one. So it's just phosphorylating, turning it on, and then these PPs are protein phosphatases that just removes the phosphate so that it inactivates itself again. So active, inactive, active, inactive. Again, it's just one step leads to the next. You can see what's a cascade. If you think about the cascading of water down a mountain, it's going from one boulder to the next. It's just rolling down, right? One step leads to the next step, leads to the next step. And in reality, this is a phosphorylation cascade, but this would be amplified. What I mean by that is this probably isn't just activating one, it's activating all kinds of these next ones. Okay. But this is a phosphorylation. What do I want you to get from this? It's activated by reception, so it's a second step, and it's just like one step leads to the next, leads to the next, leads to the next, until we get it to where we need to go so that we can create a cellular response. Now, what could be involved with this? One thing is called a secondary messenger. What I want you to know about a secondary messenger is they come obviously second. This is part of transduction. This is not reception. They're usually small, non-protein molecules. Good example is cyclic AMP. Calcium is another one. Cyclic AMP is specific. This is done in the epinephrine pathway that was mentioned earlier. It's a secondary messenger, so it's part of the transduction process. Actually, interestingly enough, this is related to caffeine too. I'm sure some of you drink caffeine in order to make your body awake. How that actually works is caffeine-containing beverages such as coffee or sodas provide an artificial way to keep the body alert because caffeine blocks the conversion that we're seeing here. It's blocking cyclic AMP from becoming AMP, and it maintains a state of activation if epinephrine isn't there. So it's like an artificial way to keep your body awake. That's what caffeine actually does. So this is just showing that. Again, this would be reception, our signal molecule bonding to our receptor. This whole part right here is actually transduction. So here's our secondary messenger. It's going to be part of transduction. Called secondary because it comes second. Reception would be first, secondary would come next. It's just part of transduction. Calcium is another example of this. It's involved in muscle contraction. It's also involved with secretion of neurotransmitters. It's also involved with cell division. So calcium is a very important secondary messenger. So again, if we look at this, we have, calcium being a secondary messenger in this process. It's coming second. It's part of that transduction piece. Other secondary messengers can be out there as well. We're not going to focus on them. They can increase calcium in the cytosol, just again amplifying that process. So here's that. Here we have our reception, our signal molecule bonding to our receptor, activating this whole part here is all transduction. So here we have one secondary messenger activating another secondary messenger. All you need to know, these are non-protein. They're part of transduction. All right, I'm going to encourage you to watch this video reviewing a signal transduction pathway, but I want to get to the response. So this is the last step in the process. Cell signaling leads to the regulation of a cytoplasmic activity. It could be transcription, which is would affect your DNA. We'll get into that in a couple units, what transcription actually does. If it's in the cytoplasm, signaling pathways regulate a variety of cellular activities. Ultimately, though, a signal transduction pathway has to lead to the regulation of a cellular activity. It's going to cause a cell to respond, in other words. This could be the opening or closing of an ion channel. It could be a change in the cell metabolism. There's all kinds of things that cells can do in response. So again, here we have back to our epinephrine. Epinephrine is the hormone. It's going to activate a receptor. This whole process is transduction. We can have a phosphorylation cascade. There might be, here's our secondary messenger, cyclic AMP. But what's the response? In this specific cell, it's the breakdown of glucose. Okay, so this hormone triggered a cell to break down glucose. That's its response. Okay. Other pathways, they may not regulate the activity of enzymes. They, like this one did, it activated an enzyme that broke down glycogen. Sometimes they don't do that. This is going to tell a cell basically to start creating a protein. So this is growth factor. Here's our reception, our signal molecule receptor. Here's our transduction. In this case, it's going to tell this cell, okay, activate this gene so that we can make this protein. This is the goal of this cell. This response would be to create a protein product. Signal pathways with multiple steps can amplify the signal and cause huge cellular responses. Okay, so first step, they amplify the signal. Second part, they contribute to the specificity of the response. How specific, these are two really good benefits, fine-tuning of a response. At each catalytic step of a cascade, the number of activated products is usually greater. That's an amplification piece. The small number of epinephrine molecules can lead to the release of hundreds of millions of glucose molecules. So one little bit of hormone can cause huge amounts of changes within a cell. Each protein in a signaling pathway amplifies the signal by activating multiple copies of the next component in the pathway. Again, signal amplification is going to not just activate one thing, it's activating all kinds of things, amplifying this process. The different combinations of proteins in a cell give a cell great specificity. I'll just give you an example. If this is epinephrine, it might cause glycogen breakdown in one. It might cause a rapid heartbeat in another. Okay, so epinephrine, if it gets to the liver, is going to cause glycogen breakdown. Going to release glucose, in other words. But if it gets to the cardiac muscle, it's going to lead to a rapid heartbeat. So this is specificity. Different hormones cause different responses in different cells. That's what this is talking about here. Scaffolding proteins, I really think of this as like hardwiring. It's not really all that of an important concept, but this just kind of, if you think about hardwiring a computer, it's all about making it more reliable, it's a quicker usually response. So that's kind of what that scaffolding does. I'm going to play this video, and that'll be it for today's notes. Oh, guess we're not going to play that video. I'm going to play the other one. This one. But Mr. Anderson does a nice job of showing this, so please pay attention.
Hi, it's Mr. Anderson, and welcome to Biology Essentials, video number 38. This is on signal transduction pathways. Signal transduction pathways are very important in the actions of cells, but they're sometimes misunderstood. So I wanted to start with a analogy. And so when Jimi Hendrix plays the guitar, he would vibrate the strings on the guitar. Those would then be transduced. In other words, the pickup in the guitar, the electric pickup in the guitar, has magnets and wires inside it. And what it does is it transduces that message of the wires into an electrical signal. It then goes into an amplifier where you can make it really, really loud. And so we can hear it. And that's what he was famous for. And so signal transduction pathways in cells do the same thing. It starts with a message, and that message is in the form of a chemical message, and then that's transduced into actions within the cell, and it also can be amplified. And so signal transduction pathways all work in the same way, and there are a couple of ways that their actions work. Sometimes they'll actually modify a protein or change the shape or the conformation of a protein, but mostly what they'll do is there'll be what's called a phosphorylation cascade. In other words, a phosphate group, which remember carries energy, will be passed off from one chemical to another until it eventually has an action. And protein kinases are important in that. And so the example that I'm going to give you today, a message comes in, a message is picked up by a receptor, and so this line we could say is the cell membrane on the outside of the cell. That message will dock with the receptor, and when it does that, in this case, it'll change the shape of that receptor. The example I'll give you is what's called the G protein receptor. Then we'll have transduction. Transduction, remember, is when we're switching that message and we're changing that from a signal message on the outside of the cell to a message within the cell. We use what are called secondary messengers. The one that I'll give you an example of is called cyclic AMP. It's a very common messenger in cells. And then that'll eventually target the cells. In this case, it's going to target cells in the liver, and it's going to make them release glucose from glycogen. And so that's kind of a signal transduction pathway. It starts with a message, and then it eventually has some kind of a target within the cell. And so let's get started. This is an animation of this typical liver cell and how epinephrine can affect it. So we're going to go through, and I'll pause at a couple of different spots and explain it. So epinephrine is the messenger. Epinephrine is going to be given off from the adrenal gland. It's going to move throughout your body, but it's going to especially affect cells in the liver. So epinephrine is going to dock with this receptor. And in this case, it's called a G protein receptor. And so the epinephrine is what's called a ligand. And so a ligand is going to be a chemical. It's a chemical that can't make its way through this cell membrane. It can't make it through this hydrophobic region, so it's going to dock with the G protein on the outside. G protein is a protein that's embedded within, it's a snaky looking kind of a protein. It's embedded within that cell membrane. So it's got a portion on the top and it's got a portion on the bottom. It's got these units on the bottom. They're called subunits. And so what happens is when that ligand attaches, in this case epinephrine, with the G protein, it causes a conformational change in that protein. So it's changing the shape of the protein. And what happens when it changes that shape is it actually releases one of those alpha subunits. And so one of the subunits, called the alpha subunit, will be released, and it's going to move to a protein just right down the way in the cell membrane. This protein is called adenylyl cyclase, and it'll make sense why it's called cyclase in just a second. But essentially what it is before the actual alpha subunit comes, it's an inactivated enzyme. In other words, it's an enzyme that's not working yet. It hasn't changed its shape, so it's actually a functioning enzyme. But once the alpha subunit is in place, it's ready to do its job as an enzyme. And in this case, what it does is it converts ATP, adenosine triphosphate, into cyclic AMP, or we sometimes call this CAMP. Now, what is ATP? We know that has three phosphates attached on the outside. It carries energy mostly in cells. We're very familiar with how ATP can be converted to ADP when it drops off one of those phosphates, but it can also drop off two phosphates, and that's what happens in this case. So now it becomes AMP, or monophosphate, but there's also a cyclic portion to it, and so it adds right where we come off of the the sugar, it's actually going to make a cyclic portion, and I'll put a picture in here of that molecule, ATP. And now we've created these messengers. Messengers are going to spread throughout the cell, and this is called cyclic AMP. And those secondary messengers, in this case, are going to target something called the protein kinase. Protein kinase, it's made up of a number of different subunits of protein, but kinase means it does something or does action. In this case, this protein kinase is going to have two catalytic subunits. Catalytic means things that are going to speed or speed up chemical reactions. And then it has these two regulatory subunits. And so as long as the regulatory portions are attached to the catalytic portions, protein kinase is inactivated. It's not going to do anything. But let's watch what happens to the cyclic AMP. It'll actually bind to those regulatory portions of the protein kinase, and it releases the catalytic portions. And so now we have this cascade. In other words, we have this cascade of energy. Those catalytic portions are going to become phosphorylated. In other words, they're going to pick up energy from ATP, and they're going to become activated. And they change now from a green to kind of a yellow activated color. They then can act on enzymes within the cell. Sometimes they'll act through a number of different molecules within the cell. In this case, it's going to drop off that phosphate to phosphorylase, and it's going to activate phosphorylase so it can release glucose from glycogen within the cell. Now, once we don't have that ligand attached anymore, we don't make that cyclic AMP, then the whole thing is going to shut down again. And so the signal transduction pathway is simply a way that we can take this message and we can move it throughout the cell and then have desired consequences within the cell. Okay, so let's do a little review. And if you've ever watched the show Dora the Explorer, there will be times where she just kind of pauses and looks awkwardly at the person who's watching the show. And so that's where you have to jump in and help a little bit. So let's do a little bit of review.
I'll spare you the Dora reference. You guys are lucky. All right. Have a good one.